Aging is increasingly understood not merely as the passage of time, but as the cumulative consequence of molecular and cellular changes, many of which are tightly linked to DNA integrity, energy metabolism, and repair pathways. Among the central molecular hubs influencing these processes is NAD, the versatile coenzyme derived from vitamin B3. NAD is essential for fueling critical enzymes and pathways that maintain genomic stability, regulate metabolic function, and orchestrate cellular stress responses. Recent advances in genetics and molecular biology have revealed how NAD interacts with key players such as WRN, PARPs, sirtuins, and other repair machinery, demonstrating that maintaining NAD availability is crucial for slowing age-related DNA damage and supporting long-term cellular resilience. One of the best-studied genetic connections to aging involves Werner syndrome, WS, a premature aging disorder caused by mutations in the WRN gene. WRN encodes a helicase and exonuclease that is essential for DNA repair, replication, and maintenance of telomeres. Loss of WRN function leads to genomic instability, accelerated cellular senescence, and early onset of age-related pathologies such as cataracts, osteoporosis, and atherosclerosis. Intriguingly, WRN activity is tightly intertwined with NAD availability. Many of the DNA repair pathways that compensate for WRN deficiency, including PRP-mediated repair, are NAD-dependent. By supplying adequate NAD, cells can enhance PRP activity, maintain efficient DNA repair, and reduce the accumulation of genomic lesions, even in the context of compromised WRN function. This mechanistic insight illustrates how NAD supplementation can help preserve genomic stability at a foundational level, potentially mitigating the impact of both rare genetic syndromes and normal aging. PARPs, poly-ADP ribose polymerases, are another class of NAD-dependent enzymes that exemplify the centrality of NAD in genome maintenance. PARPs detect and signal single-strand DNA breaks, recruiting repair machinery to damaged sites. Each repair cycle consumes NAD, linking cellular energy availability directly to DNA repair capacity. When NAD is depleted, due to aging, metabolic stress, or environmental insults, PRP activity declines, leaving DNA damage unrepaired and contributing to genomic instability. This can lead to mutations, chromosomal aberrations, and an increased risk of cellular dysfunction. Supplementation with NAD precursors such as NR and NMN replenishes NAD pools, enhancing PRP efficiency, supporting accurate repair, and reducing oxidative and mutagenic stress. This creates a positive feedback loop. Better repair sustains genomic integrity, which in turn supports cellular metabolism and resilience. Sirtuins, a family of NAD-dependent deacetylases, provide an additional layer of genomic and metabolic protection. Sirtuins regulate chromatin remodeling, mitochondrial function, stress responses, and apoptosis, all of which influence DNA stability and repair indirectly. Their activity is limited by NAD availability, so declining NAD with age diminishes sirtuin-mediated regulation. By restoring NAD, NR and NMN supplementation reactivate sirtuins, enhancing the cell's ability to respond to genotoxic stress, maintain telomere length, and coordinate metabolic signaling. Sirtuin activation has been linked to improved mitochondrial efficiency, reduced oxidative damage, and enhanced DNA repair capacity, demonstrating that NAD restoration produces systemic benefits across multiple organelles and cellular compartments. Other molecular players in DNA repair, such as DNA ligases, checkpoint kinases, and base excision repair enzymes, are also influenced by NAD levels. These pathways collectively orchestrate cellular responses to damage from reactive oxygen species, replication errors, and environmental stressors. NAD depletion compromises these processes, accelerating senescence and reducing cellular regenerative capacity. By maintaining NAD homeostasis, vitamin B3, derived precursors support the coordinated activity of these repair pathways, ensuring that cells can effectively detect, signal, and repair lesions while sustaining energy metabolism. The result is a cellular environment that is more resistant to age-related genomic instability and better equipped to maintain tissue function over time. Beyond individual enzymes, NAD impacts broader epigenetic and chromatin dynamics. Sirtuins, for example, modulate histone acetylation patterns, influencing which genes are expressed in response to stress, nutrient availability, or DNA damage. Efficient chromatin remodeling allows repair machinery to access damaged DNA more readily and supports the transcription of genes involved in antioxidant defense, mitochondrial function, and metabolic regulation. NAD supplementation enhances these epigenetic mechanisms, creating a cellular state that favors resilience, repair, and optimal energy utilization. The implications of these interactions extend across multiple tissues. In muscle, neurons, liver, skin, and cardiovascular tissues, NAD-dependent repair pathways help maintain function despite cumulative damage. For instance, in the brain, NAD supports neuronal DNA repair and mitochondrial health, which can protect cognitive function and synaptic integrity. In the liver and kidneys, NAD enhances metabolic detoxification and genomic stability, reducing organ stress and supporting long-term organ health. Even in the skin, NAD supports DNA repair mechanisms that prevent UV-induced lesions, contributing to structural integrity and reduced photoaging. These systemic benefits illustrate why NAD restoration is considered a cornerstone in emerging longevity strategies. Safety and practicality further enhance the appeal of NAD supplementation in supporting DNA repair and genetic resilience. NR and NMN are generally well tolerated, with minimal adverse effects, while niacin and nicotinamide have long histories of safe use. Their ability to raise tissue NAD levels without imposing significant metabolic burden makes them suitable for long-term interventions aimed at preserving genomic integrity and slowing age-related decline. In combination with healthy lifestyle choices, such as regular exercise, balanced nutrition, and avoidance of smoking and excessive alcohol, NAD precursors can amplify the body's natural repair mechanisms, offering a scientifically grounded approach to sustaining health span. In conclusion, NAD sits at the intersection of energy metabolism, DNA repair, and genomic stability, directly influencing the function of WRN, PARPs, sirtuins, and other repair machinery. By replenishing NAD, vitamin B3, derived precursors enhance DNA repair efficiency, maintain chromatin dynamics, modulate stress responses, and preserve mitochondrial function, creating a cellular environment that resists age-related damage. These mechanisms provide a robust, systemic foundation for longevity interventions, illustrating how targeting NAD pathways can reinforce genomic integrity, slow the accumulation of mutations, and promote healthier aging across multiple tissues. As research advances, NAD restoration is increasingly viewed not merely as a supplement strategy, but as a central pillar in the genetics of aging and the science of longevity.